you must always remember, as students and research scholars, it is not about learning, it is not about teaching, but it is about training. So the skill, whatever that you learn, it becomes quite unforgettable, and you try to practice it in your own way. So the standardization of any specific technique is an art by itself, and every time we try to standardize, we keep modulating certain steps. So that is what we mean by standardization. And probably these kind of platforms that are basically conducted to impart knowledge and skill on these areas will perhaps enable you to have these modifications and your own creativity being inculcated. So with this aim, we have initiated this process, and I would like to immensely take this opportunity to welcome all our guests, and by and large the students to have a benefit out of this program. Thank you.
Thank you, ma'am, for such a warm and inspiring welcome. Your words remind us of the importance of collaboration and innovation in tackling challenges and sustain the fast-moving world. Next, to provoke the importance and foster the scope of this seminar-cum-workshop, I am pleased to invite Professor Dinakar Rao sir from Department of Bioinformatics.
Honorable Vice Chancellor in charge, Professor Tarekar Saru, and our Dean in charge, and the Research Director, Professor Selvin, who is the pioneer in microbiology research, is also here, and our invited speaker, Dr. Arunachalam, University of New Mexico, USA, my esteemed colleagues, Professor P.T.V. Lakshmi and Dr. Suresh Kumar on dais, and off the dais my colleagues and faculties from the sister department, staff, research scholars, and participants. Very good morning. As we know, the human gut is natural habitat of a large and dynamic bacterial community. But a sustainable part of these bacteria populations are still to be described. However, the relevance and the effect of resident bacteria on host pathology and physiology has been well documented. Antibiotic resistance is a grave threat to modern medicines and global public health. The global attributable mortality of antibiotic resistance is estimated to number around 1.3 million deaths every year. This antibiotic resistance in bacteria can be mediated by a variety of mechanisms, with the most important being the prevention of access of the antibiotic with the target, or prevention of the antibiotic binding to its target, or its modification, or its degradation of the antibiotic in the bacterial system. Further, bacteria can develop resistance through mutations because of its small genome size. It can easily get mutations, and further it spread to vertically upon cell division to daughter cells. The advent of high throughput methodologies and elaboration sophisticated analytical systems have facilitated the detailed description of the composition of microbial constituents of the human gut, as never before, and are now enabling comparison to be made between health and various diseases states using bioinformatic tools and computational approaches, since large of bacteria genome is sequenced and deposited in data banks. So dear participants, in this 3-day conference, relevant talk and hands-on training is arranged. I request all students to participate in the conference actively and gain the knowledge on wet lab experiments. On behalf of the department and my own department, on my own, I thank Dr. Arunachalam, University of New Mexico, USA, for accepting our invitation. Thank you, sir. And I congratulate Professor P.T.V. Lakshmi, head the department, for organizing such a wonderful conference for students' benefit. So with this, I wish you all the best. Thank you.
Thank you, sir, for outlining the scope of our sessions. We are more excited to engage with these diverse topics and learning experiences you have detailed. Now, I would like to request Professor PTV Lakshmi, Head of the Department, to honor our Vice Chancellor with a bouquet and memento.
Very esteemed, beloved and honorable Vice Chancellor, Professor Antimicrobial, and respected Madam, Professor Lakshmi, and Professor Arunachala Muttaya, maybe he is the sole center of the focus today, and respected colleague, and on the dais as well as those who are sitting in front of the rows. So I wanted to share two of my experience. Actually, we had one of the project, AMR Watch, that is India-UK combined project funded by DBT and NERC UK. We had collaborated with Imperial College London. Particularly, we wanted to quantify, measure the outcome of industrial waste in India. Did you know India is the second largest producer of antibiotics and first largest consumer of antibiotics? We don't have any regulation in prescribing. If you have a cold, or now it is a seasonal flu, if you go to the pharmacy, they will give 2 days of doses. That will be the main reason you are developing antimicrobial resistance. So did you know we are in the 4th generation? So it usually we will say the terminology drug resistance and multi-drug resistance, extensively drug resistance. Now we are in the pan-drug resistance. Pan-drug resistance means none of the antibiotics in the pharmacy can cure the infection. So as per the CDC, there is a survey, they made it, and the survey indicates that all the population in the world, they will undergo antibiotic resistance infection as a part of their life any point of time. So nobody can escape. But the question is 2030, we are expecting millions of deaths by AMR. The biggest challenge, people are doing the work now. Instead of you are innovating new classes of antibiotics, now the discoveries must be on reversal of antibiotics. That is the frontier. Maybe it may be a Nobel Prize winning work, particularly for the students. You can think about it, because a lot of CRISPR-Cas, many tools are people are thinking, you can think about it, reversal of antibiotic resistance. Now see, my experience, maybe 1 minute I will just share, because we have made the sampling from Chennai and Pondicherry. Chennai is known for antibiotic producing, means active pharma ingredients based industry, and Pondicherry is known for not manufacturing, what packaging. They will make the different formulation industries, that is the terminology. So antibiotic formulation industry in Puducherry and manufacturing in Chennai. We made the survey, means sampling in the pollution come out of these industries. So did you know it is going to publish around, we are sitting around 600 ARGs. It means antimicrobial resistant genes. It is flowing through around us, our food system, our environment. Throughout, every day we are exposed to ARGs. So that is one of the main reasons we are going to develop the antimicrobial resistance. So it is nobody can escape. You just teach your family, your friends, your society, beware of taking antibiotics without any prescription, or it is without any bacterial infection. So this is actually, I should appreciate, actually we also made one effort, actually maybe 2 years back, same time, 1 week program with the AVMC, Harvard Medical College Hospital. This is very, very important to sensitize students, public about AMR. So I wish you all good luck. Thank you.
Thank you, sir, for your insightful and encouraging words. Your message serves as a powerful reminder of the collaborative efforts needed to be done to address global health challenges. Now, it's my great honor to introduce our distinguished research person, Dr. Arunachala Muttayan, Associate Professor of Biology at the University of Mexico, New Gallup, United States of America. Dr. Arunachalam is a well-respected figure in microbiology, especially recognized for his works on antibiotic resistance mechanisms in pathogens. We look forward to hearing your thoughts and insights. Everyone, please join me in welcoming Dr. Arunachalam Muttayan.
Dear students, research scholars and colleagues, good morning. It's a pleasure to see you all gathering here for this, in this wonderful morning for this seminar and workshop. I appreciate each one of you for being here and your commitment to expanding your knowledge and skills. Our colleagues rightly said the antibiotic resistance is a serious threat globally, and it's happening. So, but we don't have to worry about that, because to create awareness to the public and medical practitioners and research community, the World Health Organization create awareness program. So they celebrate every year. They celebrate the Antimicrobial Resistance Awareness Week in November 18th to 24th. So this year the theme of the celebration is educate, advocate, and act now. I hope after this seminar you can educate the general public, and you can act like an advocate to spread the awareness, and you can act. You can implement some research to prevent the antimicrobial resistance, and even you can develop new drugs. So, and rightly, this is the right time we organize this event to spread the awareness. And in today's seminar, we are going to talk about antimicrobial resistance in Staphylococcus aureus. So this is another, pathogen with the multiple resistance, methicillin resistance, vancomycin resistance. So we are going to discuss about that, and I hope you will learn some new information, and you will be able to use that information to spread the awareness and develop new research project to prevent the antimicrobial resistance. And following this seminar, we are going to have a workshop, RNA extraction technique in bacteria. So RNA is the starting material for transcriptional profiling experiments. So the transcriptional profiling is widely used to understand the antimicrobial resistance, to understand the mode of action of drugs, and in other molecular studies as well. So I hope you can utilize that workshop for your research projects. Some of our colleagues rightly mentioned, like collaboration is essential in our changing world. So most of the innovative ideas are coming from interdisciplinary collaboration. So I hope this workshop will create the platform for the collaboration within intra and interdisciplinary collaborations to develop new ideas, share knowledge, and develop new projects. So the purpose of our workshop is to learn about the antimicrobial resistance and gain some information, and you will be acting like an advocate to spread awareness to achieve the goal of World Health Organization. I think all the agencies around the world, they celebrate in the same time to create awareness to the public. So we are going to learn a lot of new information, and in the seminar. With that, I would like to take a moment to thank our organizing committee for their hard work. So your hard work has created, made this gathering possible. And also I would like to thank our administrative team, our Vice Chancellor and Dean, for their great support for this event. So thank you once again, and I'm excited to see you again in the seminar and workshop. Thank you.
Thank you, sir, for those insightful and thought-provoking words. Your research and dedication to tackling antibiotic resistance inspire us all, and your address has set a profound foundation for the session ahead. We are now honored to invite our chief guest, Professor K. Thranikarasu, Vice Chancellor in charge and Director of SEI and RR at Pondicherry University, to deliver the presidential address. Professor Thranikarasu is a renowned leader in higher education, guiding Pondicherry University with commitment to academic excellence and research innovation. His role as Director of SEI and RR has emphasized the university's focus on sustainable development and resource management. Under his leadership, Pondicherry University taken significant strides towards advancing research capabilities, making him an invaluable figure in our community. Sir, we eagerly await to hear your valuable presidential address. Everyone, please join hands in welcoming sir.
Respected Dean and Director of R&D Cell, Professor Joseph Selvin, and the Head of the Department, of Department of Bioinformatics, Professor P.T.V. Lakshmi, and the today's speaker and the workshop resource person, Dr. Arunachalam Muttayan from University of New Mexico, and all the faculty members of Department of Bioinformatics, research scholars and students. Good morning, everyone. So this topic of today's this 3 days workshop is on antimicrobial resistance mechanism of the gut flora. And I hope you are all in the bio background. You have some idea about the drug resistance. So anybody is here from that is 5 integrated 4 years course students also is here? No, right? No, nobody is there. Okay. I think it is they know about the resistance. So you must be also knowing that when the real antibiotic course was started with penicillin, we thought that our human being was saved. So no more the microbial attack to us. That's where we started. You know about this, correct? And then we started understanding the concept of resistance, drug resistance. There we started the issue. Still that journey is going on. There is no end for this. But even though penicillin still we are using for certain microbes, but not all the microbes as an antimicrobial agent. But why we are talking about resistance? Resistance is the normal phenomena. We also do what when we wake up, when our mother says, wake up, then in the morning from bed, then we say, yes, something serious mother is telling, we will wake up. But after some time, we don't give any response to our whoever is waking up, correct? So this person always tells this, no other things. Even students know. Students also boring with this. You have to study well. Everybody whoever meet you, they say you have to study well. This is all. You feel always bored, correct? Same thing here also. This resistance. Resistance also, the microbes also say, why these people always give this penicillin or known medicine. I know how to handle this. I know how to handle this. That's so. Please try some other things. That's the challenge it gives to us. But unfortunately, we don't get medicines very often in the market. I don't know how many of you know about the thalidomide tragedy. Can you raise your hand? Thalidomide tragedy. No? These are the reasons. Because in chemistry we teach when it comes to the stereochemistry. Earlier, before thalidomide tragedy, people used to take the drug like Ayurveda, Siddha, because these are all natural source. Even if you give and test, there is no much side effect. Like this, people used to synthesize in the industry and test directly to the public whatever disease they found. So if it clicks, okay. So whereas when it comes to the thalidomide, this thalidomide, when they tested for the vomiting sense for the pregnant woman in the first trimester to arrest the vomiting. So it clicked for certain people, certain patients. For certain patients, after during the delivery, they found out that there is the body parts were damaged. Kids with you can go and Google the thalidomide tragedy, and they found out that there is a stereochemist involved. One is S thalidomide, another is R thalidomide. One is a drug for this vomiting sense during first trimester. Another they call it as teratogen. Am I correct, madam? So it affects because first trimester only the body parts is formed for the womb. So during that step itself, it disturbs the formation of body parts. So later on only they found out that even you can see the photos. It is mostly Europe and then especially German. This tragedy happened. But when it reaches this thalidomide to America, the drug controlling agents, they said we cannot allow this drug without testing. So that is why this tragedy did not happen in US. We still now, that is the reason we still and go and check what is the approved drug in the American agency, because they do all this testing. This is good for the human when it comes to the whether the drug is safe to take, but it delays also to get the approval. The approximate I am talking approximate time to get the approval for one drug, it takes more than 10 years approximate. It may be sometime, it is, if you are lucky, it may be 2-3 years also, 4 years also, but the generally to get a drug it takes time because there is a protocol to be followed. First small molecule synthesis, then in vitro analysis, then in vivo, then the animal test, then it will go to the first clinical trial, second, like this. To get the approval, during this process the drug may be dropped also. They may find out that it is a poison or it gives side effects. But as a result, getting a proper drug for a proper disease, it takes time. That is the time our microbes get resistance to the existing drugs. So the other way, that is the reason we say in the chemistry why we are proposing new molecules for the drug. The main reason is the existing drug may get the resistance during the course of time. As you know, TB is one of the dangerous diseases. For this, the drug resistance is the main reason. Even multiple MDR TB bacteria is very famous. So the drug resistance, what I am trying to say is drug resistance is the major threat for the scientific community, and where to use, how much to use, without taking prescription also we buy the medicine and use. All those things is the major reason for the developing the drug resistance. And also in food industry, to avoid the to improve the life period. They add the sometime they add the some of the antimicrobial agents, and even they inject illegally also. Some of the chickens also they inject. That's what I come to know. Because of this, without knowing that, through food we are eating the antimicrobial agents. This is another major problem through food industry. So we have to be very careful all those things. So this is a kind of awareness. That's why World Health Organization, they said, our head of the department as well as Dr. Arunachala mentioned that. So there is a antimicrobial the antimicrobial resistance awareness week. So this is very serious threat. The one side we are worrying about the disease. There is no drug for the new diseases, and the new viruses are coming, and the evolution of viruses, trying to find out the more drugs for the or new drugs for the existing disease. But the another side, existing diseases developing the resistance. So if you see this in this war, we are not able to seeing the end. So it is, the war is still going on, and we don't see any end for this. Only, but whereas the antimicrobial resistance, to some extent it is manageable if we are really aware, our public aware about this, how to reduce the antimicrobial resistance, then we can handle this. Especially when it comes to the TB, they talk more about the antimicrobial resistance. The reason is, it is the, in India we have the DOTS program. It is 3 to 6 months continuously they have to take the medicine if they have found out the TB. But the during the 1 week or 2 week course of time where they take, they, even the doctors also, they don't tell really if you have a TB, then they may get the panic and their psychology will be affected. So they give the medicines, and then for the poor background and the education also less, those type of patients, if they take first 2 weeks, they feel that it is comfortable, and they also say they consider this as another bacteria. So generally we take 1 or 2 weeks, we stop it. But doctors specifically say you have to take at least minimum of this many months, but they ignore it. So they are, I feel comfortable why these doctors are telling this. Maybe they want to do business. So that is why they are giving this misunderstanding. Because of this, they stop taking this medicine, TB drugs, isoniazid, like this. Then once they stop, after a few months, then suddenly again the growth will be more and the severeness will be more. Again they go to the doctor, then the doctor says, why you have stopped? It was good, that's why we stopped. Then when they give again the same medicine, it generally doesn't work. During that time it develops the resistance. So that's why they say there are different in TB different type of drugs. If isoniazid doesn't work, then they go to the second line, first line drug, second line drug, like this. So the antimicrobial resistance, to some extent we can control. Like if you use it with proper understanding, if you don't abuse this using often, then we can reduce the antimicrobial resistance. So awareness is one of the criteria to reduce the antimicrobial resistance. So when it comes to the mechanism, it is only whatever I talk is the general understanding. But when it comes to the mechanism, it is really research. We don't teach mechanism to the public, but it is only for the reason, only for the researchers to understand this. That is the reason this present workshop is going to talk about this. So how the mechanism in my lab also, I first when they come for the, because basically students are chemists, and they synthesize novel molecule and they test for the submit for the test for any in vitro analysis. Once they come with the result, then second stage is I used to ask, do you have any idea about what is the mechanism of action, whether the existing mechanism or the new mechanism. But it is not easy to find out the mechanism. There are different ways to find out the mechanism for the different diseases. But this is the very complex mechanism, and where researchers can put our understanding about these different ways to come out with the mechanism. That is the our resource person from New Mexico, Dr. Arunachalam Muttayan, is going to talk about this. So this will be very useful for all the students, and I came to know that he is also going to give you the wet lab training also to how to the separate RNA, and how to the also stabilize this and use it for the easily degradable RNA and all other things. So this is going to be really useful for you, and there is no in biology there is no such thing as you can survive with only wet lab experience, and also there is no thing that you can survive with only computational biology. It should be go in hand in hand. So it is the really good opportunity for you to be in this department, having experience both in the wet lab as well as the computational. Is a very rare mix, which is very good for the future researchers. And I also discussed with your teachers. This year there were many awards, Nobel Prize for the artificial intelligence, computational biology especially for the. So madam also was telling during 1990s or 80s, correct? 80s when they were initially starting about the computational biology, all those things. The center stage which was taken by wet lab people, they don't even recognize this type of work. Now you have taken the center stage, and they are viewing you as the how these people are going to give us the solution for the problems. So this is the scenario, you can see how fastly it's changing. So this is a very good opportunity for all the bioinformatics students to have more and more the wet lab as well as computational experience, and with the advent of AI, it is going to be very useful for you as well as the community to get more benefit from bioinformatics. In this aspect, I am really thank the Department of Bioinformatics for organizing this seminar cum workshop on antimicrobial resistant mechanism of the gut flora. So thank you, thank you once again. Thank you.
Thank you, sir, for your inspiring address. Your message reaffirms our advancing research in antimicrobial resistance and addressing global health challenges. Now, to bring this inaugural session to a close, I would invite Professor M. Suresh Kumar, Department of Bioinformatics, to offer the vote of thanks.
Thank you. I thank Professor Dharanikarsu, Vice Chancellor in charge, in his busy schedule. So he finds some time to come to our department to start the seminar. And I thank the registrar and the finance officer for releasing the money, and also thank the funding agency, DBT, for giving us the money to conduct the seminar. And then I thank our technical staff and also the people from media and communication who are frequently coming here and taking care of us in propagating the seminars and all the things, whatever we are doing. So I thank our faculty and also the faculties from our life science department who are spending some time here. So thank you once again. So I thank the chief guest who is from University of New Mexico, Arunachalam, to teach our student all the techniques so that they get the benefit out of that. Thank you once again.